This is part 2 of cell structure and function. In part 1, I went through the proteins of the cell membrane and how they function and what they do. In this video, I'm going to go through the cell organelles located within the cell. The first structure we're going to look at is the nucleus. The nucleus is the brain of the cell. It contains chromosomes. It contains chromosomes that contain DNA, which contain the genes, which are the instructions for the proteins of your body. So it contains kind of the blueprints for all the proteins that you need within your body. Again, it's the brain. It controls function and controls what the cell is doing. When we get into cloning, we'll talk about how you need the nucleus. You need the nucleus in order to clone an organism. Once the blueprints are located in the nucleus. Now, if you had blueprints, you wouldn't want them to leave that protected environment. So what happens is, and we'll talk about this later, is those blueprints are copied, the gene is copied, a copy of it is sent to the ribosomes, and then at the ribosomes what's going to happen is they're going to take those instructions and it's going to put amino acids together to produce a protein. In the last video we talked about the role that insulin plays in helping maintain blood sugar. Well, there's instructions for that are located in the nucleus. A copy is made, sent to the ribosomes where the insulin will be put together. Now, endoplasmic reticulum. There are two types. There's the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The difference is rough endoplasmic reticulum contain ribosomes where those proteins are made. So in rough endoplasmic reticulum, when the proteins are made on the ribosomes, it goes into the endoplasmic reticulum where that protein is then shaped and modified and also checked to make sure that it's been put together correctly. If it hasn't been put together correctly, then the ER can destroy it. We want correct proteins because the shape of a protein, which is determined by the amino acids, if that shape changes, then it's not going to function. There's also smooth ER, which deals with the production of phospholipids and so on. So again, if mistakes were made during protein synthesis, the protein is destroyed. So again, rough ER has the ribosomes, smooth ER does not. So smooth ER is detoxification, so the breakdown of molecules, production of phospholipids, and so on. From the ER, the Golgi apparatus, or the protein, is going to go to the Golgi apparatus, and what it pretty much does is it further modifies and packages the protein for delivery. So what you see here, these little balls, they actually contain proteins. So in the case of a pancreas cell, we have the DNA instructions in the nucleus, the insulin is put together through ribosomes, checked and modified in the endoplasmic reticulum, then shipped to the Golgi apparatus, which is then going to further modify it and package it. And then what will happen is, imagine one of these little balls contains insulin in it. It will travel to the cell membrane where it will be released into the bloodstream and then bind to receptors on cells and influence blood glucose level. Golgi apparatus, this just shows you here kind of an animation. You think of the yellow as the protein. It's stored in the little ball, part of the Golgi apparatus, and then transported either within the cell, if the protein is needed in the cell, or out of the cell, like we talked about in terms of insulin. All this takes energy. Mitochondria, what it's going to do, it's going to make that energy. And it makes that energy in the form of what we call ATP, adenosine triphosphate. So all this stuff going on in the cell, it takes energy, just like it takes energy for you to move and do your stuff. So the mitochondria makes this energy that the cell then uses to carry out all these functions. If you look at a muscle cell of a highly trained athlete, you will see they have more mitochondria because those muscles are constantly working. In order for them to contract, they need ATP. So what happens in a trained individual, they develop more mitochondria, they produce more ATP, they have the ability to produce more energy. This is the powerhouse of the cell. You also sometimes hear it as the mighty mitochondria. Lysosomes are kind of the digestive system of the cell. In order to make energy, the mitochondria needs energy. The lysosomes can break down molecules so that the mitochondria can use it as energy. They can also act as a recycling bin kind of thing. They contain enzymes that can break down cell parts that are not functioning or that are too old, and they also in some cases can actually digest and break down the whole cell. Centrioles. One of the things cells have to do is they have to divide. Not all cells can do this, but the process of cell division is called mitosis, which you'll do in a future module. When one cell becomes two, these centrioles are going to help with that function. They will help pull the cell apart, and more importantly, to make sure that each new cell is going to get a correct copy and number of chromosomes or DNA. Because if you're going to make two cells, you want them to have the same instructions, and so the centrioles are going to assist in that process. Microtubules can be used for several things. The cytoskeleton, it's going to give the cell shape. Centrioles are example of microtubules. Microtubules are built and broken down to transport things. The reason I use the train sound is because what will happen is if a protein or something has to move in the cell puts these microtubules down like a railroad track, and then the protein or whatever needs to be transported can follow those railroad tracks to where it needs to go. So things are not just floating randomly. Some things are, but not things that have to be moved to a specific part are being transported through these microtubules. So whenever things move around, or when you give the cell shape, then you're dealing with what they're called microtubules or cytoskeleton. At one point, we'll talk about neurons or brain cells. They have a very unique shape to them, and this unique shape is produced because of this cytoskeleton or these microtubules. If that shape within a neuron changes, what happens is the neuron can't do its job, and that's one of the things we see in Alzheimer's brains, is that we get these things called plaques, and what happens is the neurons don't have their normal shape. So there you go. Another reason I use the train is, if you don't know this, is that Vincennes has the most amount of railroad crossings per capita. So if you don't hear a train when you're in Vincennes, something's wrong.